The overall goal of these procedures is to produce a fusion protein of the anti-inflammatory cytokines IL-4 and IL-10, control its quality, and evaluate its functional activity and analgesic properties in murine models of chronic inflammatory pain.
These detailed methods aid in developing novel biologics and test their quality and functionality. The advantage of these techniques is that you can produce sufficient amount of recombinant fusion proteins, which enable its assessment for its analgesic properties in vivo.
To evaluate the therapeutic potential of the new developed fusion protein, it is important to have a well-characterized batch and to use a variety of behavioral tests.
The implication of these methods is that they provide insight in highly needed new therapies for chronic pain, but also extend the potential towards other inflammatory diseases.
Generally, scientists new to these methods will struggle to be able to perform all procedures shown here, since expertise in different fields is required.
After subculturing 100 milliliters of HEC293F cells in 500 milliliter Erlenmeyer flasks, according to the text protocol, add 6.6 milliliters of DNA transfection reagent mixture to each flask of cells. Culture the cells at 37 degrees Celsius, 8% CO2, and 95% humidity on an orbital shaker, rotating at 125 RPM for 3 days. Then, harvest the culture supernatant containing secreted IL-410 fusion protein. To carry out protein purification, pass 100